Study the viability of some bacterial species isolated from contaminated soil on the manufacture of nanoparticles, provided by Dalia Abdullah Muhammad. Introduction. One of the most important fields for researches in nanotechnology is biosynthesis of metal nanoparticles due having numerous applications in various fields. Therefore, the study in this field were turned on to use biological approaches because these nanoparticles having the ability to synthesize nanoparticles in an eco-friendly method and having significant advantage compared to other processes. Bacteria is the most vulnerable between microorganisms because it is relatively easy to handle and can produce biocomponents like enzymes and proteins that form bioreducing used as cabin agents which make nanoparticles. Material and method. Isolation bacteria from soil sample contaminated with generator oil was purification and identified biochemically and molecularly based on 16S RNA gene sequence analysis. Silver nanoparticles was synthesized by adding 50 ml of suspension for selected bacteria in this study to flask containing 50 ml of 1 millimolar IgNO3 solution and incubated in dark conditions. Initial detection of IgMBA synthesis was noted through color change of reaction mixture, and it analyzed its optical characteristics by UV visible spectroscopy and investigated the functional group by FITR. The antibacterial activity of synthesized IgMBAs against some pathogenic bacteria like E. coli and Staphylococcus aureus was investigated by disk diffusion method. From the observation of inhibition zone appeared around the well can determine the antibacterial activity against some pathogenic bacteria. Research and discussions. The isolated bacteria from soil were identified biochemically as gram-positive bacilli forming spore and identified by 16 RNA gene sequence as Bacillus paramyxoides, Bacillus subtilis, Bacillus cereus, and Bacillus with many, illustrated in Table 1. This figure illustrated the formation of silver nanoparticles by supernatant of bacteria selected in this study. Tube number 2, when reacted with AgNO3, tube number 1 was observed by color shift of reaction mixture from pale yellow to brown or yellowish brown, tube number 3. Figure show the measurement of UV visible spectroscopy can observe it absorption peak located between 404 to 444 that suggests the presence of surface plasmon resonance of silver nanoparticles. Illustrated the FITR analysis for silver nanoparticle solution that showed the presence of band located in the region between 3344 to 3310 that corresponding to amide group confirm the presence of a protein in the sample. Also, the presence of band located in the region related to the carbonyl amide and nitrate. These groups possibly can participate in the fabrication of silver nanoparticles. Table illustrated the antibacterial activity of silver nanoparticles synthesized by supernatant of Bacillus with many, Bacillus cereus, Bacillus subtilis when reacted with 3 millimolar IgNO3 was exhibit highest efficiency against Staphylococcus aureus comparison to E. coli. This figure illustrated the antimicrobial activity of silver nanoparticles synthesized by supernatant of Bacillus strain against Staphylococcus aureus and E. coli. We conclude from this study the possibility of bacteria isolated from soil contaminated with motor oil to reduce silver ion to silver atom by producing some compounds such as a protein and enzyme to form nanoparticles. That is showed more effective against Staphylococcus aureus than E. coli when the supernatant of Bacillus subtilis, Bacillus cereus, Bacillus weidmanni reacted with 3 millimolar AgNO3. Thank you for listening.